Today's topic is about brain development in the embryo. So, during the brain development, there is a cerebral cortex formation occurs, and that is responsible for most of the important brain functioning. So, this cerebral cortex development is occurred, or before the cerebral cortex development occurs, there is a cortical layer formations occurs, and this cortical layer development is been enhanced when this Trkb that are present on the cell surface interacts with this EGF. Thereafter, that will uh, triggers more Trkb on the cell surface, and this Trkb then helps in this cortical layer development. However, how the intercellular process occurs is not been known. So this paper, the paper that I am showing, the graphical abstract of the paper that I am presenting, discuss that it's the Calnexin that plays an important role in this whole process, that is expressing Trkb on the cell surface, and that helps in this healthy cortical layer formations that leads thereafter leads to the cerebral cortex development. So what is the normal process? Normal process is that the EGF interacts with this EGF receptors, thereby triggers the more TRKB to express on the cell surface. However, when this EGF is not present, what people have seen is that, or the EGF is not present, or EGFR is defected. In any of this case, what people have observed that there is a intracellular upper intracellular TRKB that are formed will be diverted to this ERphagy or endoplasmic reticulum phagosome. And how this happens? This happens because of the overcrowded of the endoplasmic reticulum, and that will leads to the more unfolded structures, more unfolded proteins formation of this TRKB, and that will divert them towards this phagosome through the ERAT pathways or phagophore. And this how the ER functioning that we have discussed many times in our previous videos, you can refer to those videos. So this is happens when the EGF is not present. So when the EGF is present and Calnexin is present, how the normal process occurs, that is EGF interacts with this EGFR receptors, that will leads to this MAPK signaling pathways, and that will cause this more of this TRKB to be translocated to the cell surface, and thereby that helps in this cortical layer development. And in that case, since the amount of TRKB within the endoplasmic reticulum remains within the threshold level, that the ER stress is not triggered, thereby this ERphagy will not occur to a greater extent. And when they check the cells in the absence of Calnexin and in the absence of EGF, what they find out is that there is neither ERphagy they have seen, or nor the expression of this TRKB on the cell surface is increased. So this will lead to the increased TRKB within the increased TRKB within the endoplasmic reticulum, or within the cells within the endoplasmic reticulum, or within the intracellular parts of the cells, and thereby causing problems in the cerebral cortex development. And it will also affect the other functions because of being overcrowded in the endoplasmic reticulum, that will cause the more unfolded protein formations, and that are not being taken care of as the chaperone, that is Calnexin, is not present within the endoplasmic reticulum. So in case where EGF is present, however there is no Calnexin is there, in that case what will happen, this EGF will trigger this EGF will trigger the EGF will trigger this TRK to expressed. EGF will trigger the phosphorylation of the TRK to expressed to express or translocate the TRK into the cell surface receptors. However, since the calnexin is not there, so these proteins will not be able to form their native structures, and because of non-native structures, there is a impair in translocating those translocating those TRK into the cell surface of the into the cell surface on the cell surface, and thus it will affect since there is low amount of TRK expressed on the cell surface, it will affect the cortical layer development and thereby the cerebral cortex development, and the ERphagy will also not occur, so that will cause with time increase in the PTRKs in the intercellular parts of the cells, and that will cause the more unfolded protein formations, not just only for this TRK but for the other important proteins also. So it will cause the other problems along with this impair cortical layer development. So I hope you understood a brief idea what this paper is talks about, that is how the calnexin helps in this cerebral cortex development. So calnexin is a chaperone, intercellular chaperone that helps in the protein folding, and thereby it will lead to this native post-translation modification, which is essential for the proper functioning of the cells. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.